Thank you very much. I'm happy to be here, and let me greet all of you. Also, I'm here, as said, instead of Lisa Gensel, who apologizes she couldn't make it here. So we are in the success stories section, and I'm gonna introduce you the FANS-Kavli network of excellence, and this is indeed, I think, a very good example of a fantastic success story. So let's decipher the name first. It is conceived and serving FANS, the Federation of European Neuroscience Societies, and it is funded by the Kavli Foundation, and it is so members of this group are selected solely based on scientific achievement, hence the name excellence, and it is a network of 30 young PIs in Europe, and it was born in 2014, so it is 10 years old now, and I think now we can say that it did indeed brought about a lot of impact. So first, I introduce the network a bit. So who are we? So this network is always 30 people of young PIs, so basically middle career neuroscientists in Europe, and there's the mandate is for 4 years. I was a member of this network from 2016 to 2020, so I was in the second cohort, and that also means that there's a growing network of alumni. So alumni kind of provide a support network for both the active scholars in the FANS-Kavli network and also for FANS itself if they're needed. And it is a very diverse network. Intentionally, it is across disciplines, really covering the entire breadth of neuroscience, and that is a very exciting experience I'm saying this from first-hand experience, and it is a very fostering environment for scientific exchanges. But so this network does actually many other things. Not only scientific exchanges, besides science, which is, I would argue, still the most important function of this network. So integration of European science is very important if we want impact. I think the previous session really convinced us about that, and there has been many exchanges within the scholars, scientific exchanges, and there are 2 meetings a year. One is entirely dedicated for science. The other meeting is dedicated for other activities of the network, and the network has been very active in both mentoring young PIs, but also scientists at different levels, and they're also very active in science communication and outreach. So, for instance, I was on the board of governance for 2 years of the FANS-Kavli network, and I was the board member responsible for outreach, and many of the outreach activities are linked to the biannual FANS Forum, which is the largest neuroscience meeting in Europe. And we organized, for instance, many public talks. There were 10 public talks in Berlin that year in parallel to engage communication with the society. And also the network is very active in science policy. It's usually giving advice to FANS, but also to other liaison bodies. And so since we are 10 years old now, we're looking a little bit back to assess the achievements, and really there has been many meetings and exchanges, and this did indeed result in significant impact if we want to measure the actual scientific output of the network. But actually, for instance, for me personally, I felt it was very helpful that when I was returning from my postdoc period from the US, I quickly realized that actually the US network is very distinct from the European network. So all my network was located in the US, and I was kind of cut from it. And I realized that I know very little of the leading figures of European neuroscience. So this network quickly created a very good network for me within Europe, and I think that's kind of important for many young leaders of European neuroscience, as many of them actually return from the US, and many of them are US trained, so they do need a European network. And as I said, the network was also very active in mentoring. One aspect I'm particularly fond of. We introduced a child care grant at the Defense Forum of Berlin, where people could apply for relatively flexible funds to help them to attend the forum while raising children. And the network also runs prizes for successful and influential PhD thesis, and also mentoring prizes for more senior neuroscientists. And as of recent, the network also has a prize for achievements in equity and diversity. And as I already mentioned, the network tries to engage as much as it can with public at all levels, from high schools to general events. This is something I particularly find important, and I also can very much enjoy. And a large part of the network is also very active in science policy, and trying to influence how science funding is shaped in Europe, in particularly for neuroscience. And so there are two particularly important hot topics I wanna touch upon next. In terms of what is important and what will be important in terms of our decisions for Europe and European neuroscience in the very near future. And the first one is about animal research. So there's ongoing thoughts whether and how much animal research we still need, while we still have the goal of trying to understand and get better cures for brain-related diseases. And I think from within neuroscience, it's very clear that probably the moment is not yet here when we can completely phase out animal models for neuroscience. I actually work both in animal models, and I also work with human patients. Actually, with the animal studies, I'm often criticized whether it has a human relevance, and for the human studies, studies, I'm often criticized that there's little insight about the actual mechanisms, which to me means that we do still need both. I think it's gonna be important to understand. Of course, doing science and the way we do science is a society decision. So I'm not just talking from a selfish point. I'm also a mathematician by training, and I think I would be very happy actually to switch to theoretical neuroscience. I think I would get rid of many of the difficulties and burdens I'm facing now. But looking this from the society point of view, I would argue that we're not yet there where we can completely switch to only human and theoretical experiments if we still want to make progress with these diseases we are facing. And of course, it doesn't mean that it's not worth it to think ahead. And I think it's generally very important to realize that science is not always very fast for many very obvious reasons. But maybe less obviously, that also means that we need somewhat longer-term plans. So it is worth thinking about how we wanna change the balance between, say, different model organisms, which is already happening. Of course, I would like to point out that that is an effort, and of course an effort that needs a lot of thought and also needs some money. So this type of transition should be well thought for, well planned for, and well funded as well. And so this segues me into my other topic of science funding in Europe. So I'm now presenting a success story of which is increasing Europe's competitiveness in science, which is very important. And it's very important because we're not doing very well in comparison to the US, and also probably we're not gonna do very well in comparison to say China if we just continue our path. And so science is a long-term investment. Actually, it actually has the very same logic as any complex investment. So from the hindsight, it's very easy to say that you just had to buy Tesla bonds, right? It's easy. But as an investment, you want to have a diverse portfolio where your outcome will be not just a sum in your net bank when you sign in, but it's going to be also, the outcome is also the quality and the length of your life. But it's still a complex investment, and that needs money. The more you put in, probably the more you get out. And Europe has a very good tradition of science, which will make it seem like it's running on its own, and it has some inertia, so it won't disappear tomorrow if it's not well funded, but it will slowly lag behind. And if we realize this too late, then by the same mechanisms, which are not always super fast, it's going to be at that point very hard to bring it back to the top. So I will end with this, and I would actually thank this opportunity I got from this network, and I would want to advertise it also for young neuroscientists to think about applying to this network. It is what the biggest thing what it gives you is the network, so a very strong group of scientists in Europe which can help you with your science, with your grants, and all sorts of other activities. Thank you very much for the attention.
Thank you very much, and also I would like to thank you very much for stepping in for Dr. Genzel and sharing these very interesting projects with us. And our next speaker is Dr. Karolina Pircs. Please also welcome her on stage.
Thank you so much. So I'm going to switch gears a little bit. Welcome, everyone. First of all, I would like to thank the organizers for the possibility to talk our research here today, which is about direct neural conversion, which is a new preclinical model for studying aging and age-related neurodegenerative disorders. So, as we have heard today and yesterday, dementia worldwide is becoming a more and more bigger problem on the society, on the healthcare system, and on our economy. Currently, more than 55 million people are affected by dementia, but this number will almost double every 20 years. And often there are most known neurodegenerative disorders behind dementia, like Alzheimer's disease, Parkinson's disease, Huntington's disease, or due to Bruce Willis, now we know frontotemporal dementia, but there are obviously other less known neurodegenerative disorders. In a population of 50 million people, about 1.5 million people will be directly affected by neurodegenerative disorders. And as our population ages, this number will obviously rise. But there are obviously people that are not directly, but indirectly affected, like the caretakers, the social workers, the family. And why is this so important? Because there's no disease-modifying drug treatments available for these diseases. Why is that? Why didn't we solve this problem? So the average human brain is obviously very complex with like 86 billion neurons, all of them having like 7,000 synaptic connections with other neurons. So finding out what's going wrong in this, in some cells is very much like trying to find a very small needle in a very, very big haystack. And the other problem is that to develop a drug is around 10-15 years in time, but it's not just the time that goes into it, but also it costs quite a lot. So it's like something from $500 million to $5 billion. And obviously, if we can ensure in any way the success and increase the success of these neurodegenerative disorder trials, that would be very, very useful. So the question is that how do we test a drug before use in humans, and can we predict success with today's models? As you can see here, these are the most commonly used models in the lab to study aging, human aging, and age-related neurodegenerative disorders. Obviously, they all have pros and cons. Animal models are used, and they're extremely useful, but obviously a mouse or a rat brain differs a lot in size. It's less complex, it has a shorter lifespan, and important to highlight that Alzheimer's, Parkinson's, or Huntington's disease are human-specific diseases. Post-mortem human brain tissue is extremely valuable, but very limited in availability, hard to standardize it. Shows us only a snapshot of the disease, where many times the neurons that are a main interest are already degenerated. Cellular models like embryonic stem cell-derived neurons and the induced pluripotent stem cells have completely revolutionized the aging field and the neurodegenerative diseases modeling. As they are human origin, induced pluripotent stem cells are easily accessible, but we should not forget that due to the dedifferentiation, the rejuvenation phase and epigenetic erasing is happening. So these cells, these neurons, for example, lose the aging signature of the donor. So during my postdoctoral years at Lund University in Sweden, I have been involved in a project where we developed and used a new neurodegenerative model, where we used cells of human origin that were patient-derived and disease-specific neurons that most importantly maintained the age. We were capable to use many patients at the same time in a reproducible, robust way. Relatively quick, in 4 weeks, we had the neurons in a relatively cheap way. So we started to work with patient-derived induced neurons. First, you need to harvest skin biopsies, either from healthy donors or from patients with different neurodegenerative disorders, age-related disorders. Then you start to culture the fibroblasts, the skin cells, and then using viral vectors, we transdifferentiate the cells, directly reprogram them. This is important that there's no stem cell phase, no neuroprogenitor phase. This is transdifferentiation, meaning that induced neurons contain the genetic, but most importantly, the epigenetic aging signatures of the donor. In 2017, together with the Palmer group at Lund University, we have published an all-in-one lentiviral vector which was capable to efficiently reprogram adult human dermal fibroblasts into neurons. These neurons expressed mature neuronal markers like MAP2 and tau. They also resembled a proteomic and a transcriptomic profile of neuronal cells. And since 2017, we were capable to reprogram over 150 different donors from different clinics in Sweden, in Cambridge, in Italy, and also now at the Semmelweis University and Szeged. We were capable to reprogram cells from healthy donors, sporadic idiopathic Parkinson patients, familiar ones, Alzheimer patients, Huntington patients, and frontotemporal dementia. Here in this next image, you can see 3 different healthy controls and 3 different Huntington patients' skin biopsies, skin cells, converted into induced neurons. You can see with different magnifications that the neurons express mature neuronal markers, shown with red and green, MAP2 and tau, and also you can see the neuronal morphology of the cells. So let's see this, how efficient is this technology in numbers? So from 50,000 fibroblast cells, we get over 20,000 neurons, and this conversion efficiency is good enough to perform various different downstream analysis using these cells. We're capable to do immunocytochemistry combined with high content automated screening microscopy, Western blot analysis, patch clamp electrophysiology, mass spectrometry, bulk and single-cell RNA sequencing, and DNA methylation. So here you can see that induced neurons, they do keep the aging signature of the donor. By performing DNA methylation array, by using an algorithm, we can define the biological age of the cells by using the Horvath epigenetic clock. Here you can see that when we compare the biological age, the Horvath age, with the chronological age of the controls, we have a very high correlation. While if we compare neurons from Huntington patients, we see an accelerated aging, a significantly older these neurons, and this is the same like we can see in post-mortem human brain tissue. The other methodology that I would like to highlight is that we are capable to use high-content automated microscopy to analyze these induced neurons. So basically, you put the cells into this microscope, which can take hundreds and hundreds of images in a completely automated way. It's connected to a software which can analyze thousands and thousands of different neurons on a cell-by-cell basis, based on intensity, shape, and other things, to define conversion efficiency, purity, neuronal morphology, and different expression of markers within. We can also look at live cells. So basically, that means that the ion-based technology allows us for drug screening, as it's compatible with the high-content screening, and also we were capable to downsize our methodology from 24 well plates to 96 well plates. But now, the outstanding questions are, can we identify drugs that reverse age-related disease phenotypes? And does such drugs work better in humans than those identified in other model systems? With the generous support of the NIDIO, we are currently developing a new preclinical drug screening strategy for studying age-related diseasing using these induced neurons. So basically, taking different patients with different cognitive disorders, taking their fibroblasts, making induced neurons, and building a drug screening platform as a new preclinical method to to perform different drug tests. So many of our projects are focusing on the development and the fine-tuning of this drug screening platform by doing reverse translational studies, where basically we know a drug works, but we want to find out like how and why it works actually, where we want to validate something, like for example our recently published paper together with the KI, where they had clofamizine working, showing positive effects in Huntington's disease in C. elegans, and then also in flies, and also in zebrafish, and different cellular models, and we verified it, validated it in our induced neurons, Huntington-derived induced neurons. We also use this model for drug repurposing, drug screening, precision medicine, and neuronal rejuvenation. And lastly, we also used and produced, by using induced neuron, the first ever phosphoproteomic data set from human Huntington's disease-induced neurons, and found very distinct post-translational dysregulation present in Huntington's disease. And our goal was obviously to find new potential therapeutic targets in the disease, and also highlighting the importance of studying post-transcriptional modification. And lastly, I'm going to show that we also use this all-in-one vector that I showed you beforehand to try to develop novel models for regenerative medicine by using an ethically more easily accessible cell type inside the dental pulp, inside the teeth of milk teeth and wisdom teeth. And basically, we are directly reprogramming dental pulp stem cells into neurons and characterizing them and also transplanting them. So altogether, I have shown you today that ions are an excellent model to study aging, human aging, and late onset neurodegenerative disorders, as they retain the aging signatures of the donor. It allows for the analysis of neurons obtained from many, many patients. They represent the majority of patients. It's a robust protocol compatible with high content screening, which allows for drug screening, and therefore they offer a new possibility to identify potential novel therapeutic targets. And with this, I obviously reached the most important slide of all. I would like to acknowledge my group here at the Semmelweis University and Lund University. I would like to thank our wonderful collaborators, our funding, and thank you for the attention, and I'm happy to take any questions in the break. Thank you.